We all know that plants cannot perform photosynthesis in the dark, but every day as the sun rises, the light brightens up the sky. As soon as the light strikes the leaves, the sleeping leaf cells wake up without any delay, as the task they have been assigned is an important one. Throughout the day, these cells will harness the energy coming from the sun and prepare the food to feed all the hungry cells of the plant. But not like this. It becomes possible as these cells have a very special structure called chloroplast. Chloroplast is a site where the process of capturing sunlight and converting it into food occurs. So sometimes it is also referred to as the kitchen of the cell. The fluid enclosed by its membrane contains many substances like its own DNA, RNA, ribosomes, and many proteins and enzymes. But none of these is capable of capturing the light energy. Capturing the light energy requires pigments, and the pigments are not present in the stroma. Instead, these pigments are associated with the internal membranous structures, Grana, in the form of groups called Photosystems. Each photosystem consists of 200 to 300 pigments called accessory pigments and a special type of chlorophyll called reaction center. There are two types of photosystem, Photosystem 1 and Photosystem 2, named on the order of their discovery. The composition of accessory pigments and the reaction center chlorophyll molecule differs in both types of photosystem. In Photosystem 1, the reaction center is P700, and in Photosystem 2, it is P680. You can refer to the article linked in the description below for more detailed explanation. Beside the photosystem, the Grana also has some other proteins and enzymes like the cytochrome complex, which is made up of two cytochromes, cytochrome B6 and cytochrome F. Enzyme ATP synthase, responsible for producing ATP, is also present in the Grana. Now let's understand how the light energy is converted to ATP. When light falls on the photosystem of Grana, the many accessory pigments present in each photosystem absorb light of different wavelengths. All the pigments now transfer this energy to the reaction center of photosystem, and as a result, the reaction center acquires sufficient energy and starts to release electron. The electron released from the photosystem 2 are captured by an electron carrier plastoquinone and transferred to the cytochrome complex. While transferring electrons, the plastoquinone also transfers protons from the stroma of chloroplast to the lumen, thus acting as a proton pump. From the cytochrome complex, the electrons are transferred to the photosystem 1 through another electron carrier, plastocyanin. The electron released from the photosystem 1 are captured first by iron sulfur protein and then by ferredoxin, and finally transferred to an enzyme NADP reductase located towards the stroma side of the membrane, and it does what its name suggests, that is, reduces NADP plus to NADPH. But the continuous loss of electrons from the photosystem 2 creates a deficiency of electron in the photosystem 2 reaction center. This deficiency of electrons is overcome by breaking water molecules in the lumen by the oxygen evolving complex associated with the photosystem 2. The resultant proton gradient created by a continuous flow of electrons from photosystem 2 to photosystem 1 provides sufficient proton motive force to make ATP. The ATP and NADPH thus formed are used for producing glucose in the stroma, which will be covered in a separate video. These processes continue throughout the day, and after working tirelessly, the leaf cells get their well-deserved rest to be ready for the next day.